Hello and welcome to the CARIS webinar series. I'm John Bohl and I'm today's session moderator with technical support provided by Liz Mueller. Thank you for attending. I'm really pleased to have been asked to introduce today's presentation because we have a special guest speaker, Dr. Judy Westrick of Wayne State University. As you will learn, Dr. Westrick is a water industry expert with a deep understanding of drinking water treatment and its chemistry, and of one of the most important topics today, cyanobacteria and cyanotoxins. Dr. Westrick has been working hard to try and teach us here at CARIS about this issue, and I can tell you that today you're in for a treat, so get ready to learn. But first, let me start with some important information about our webinar. Today's webinar is being recorded, and it will be available on demand after the live session. All attendees will receive a follow-up email after the webinar is over containing a link that will give you access to the recording. We encourage you to share this within your organization and your network of friends and coworkers. If you're interested in receiving a PDF of the presentation slides, please contact us directly. To ensure the best audio quality, we have everybody in listening mode only. And if you have questions during the session, we ask you to submit them throughout the presentation using the question pane on your GoToWebinar screen. We will be answering as many questions as possible at the end of the session as time permits. If we do not get to your question, we will follow up with you directly after the webinar using the contact information that you provided. We have also included literature pieces that appear in the handouts section of the webinar screen. They can be opened, downloaded, and saved while the session is in progress. Our 2017 webinar series is underway, and here's a look at the list of webinars that we have previously hosted and are available for viewing on our website. Also, we will announce our second half of the year webinar series on our website, social media, and through email blasts. If you currently do not receive our email blasts and would like to join, please feel free to contact us about that. Here are a few more ways to find us. Visit our website at cariscorporation.com for product and technical information, webinar links, webinar schedules, and more. We have included our sales and marketing email addresses as well. Follow us on LinkedIn, YouTube, Google+, Facebook, and Twitter, where we frequently post technical articles, upcoming events that we are attending, webinar schedules, as well as recordings, and much more. So at this time, I'd like to take a few moments to introduce our speaker, Dr. Judy Westrick. Judy Westrick graduated from Wayne State University and earned a PhD in physical organic chemistry from the University of Colorado Boulder in 1989. Since her PhD, she has performed research with several interdisciplinary groups, such as the Institute for Behavioral Genetics, the US EPA, and the American Water Works Association. In 2001, she joined the faculty at Lake Superior State University. She founded and directed the Lake Superior State University Environmental Analysis Laboratory, a self-supporting laboratory used to fund undergraduate training and research, as well as K-12 camps and professional workshops. During her tenure, she mentored 81 senior research projects, advised the chemistry club, and was a key player in the Lake Superior State University Chemistry Department becoming ACS accredited. As a leader in cyanotoxins and harmful algal bloom research, she has managed numerous occurrence studies, determined cyanotoxin susceptibility to drinking water treatment products and processes, developed analytical methodologies, and validated commercial analytical products such as cyanotoxin ELISAs. Dr. Westrick has organized and served on several expert panels, reviewed grants for numerous federal agencies, written reviews, edited a special edition of Toxicon. As the new director of the Lumigen Instrument Center at Wayne State University, her goal is to provide professional and high-quality research and instructional support to Wayne State University researchers, as well as to the greater Detroit and Michigan communities. And with that, we'll turn the controls over to Dr. Westrick. Judy, take it away.
Okay. Well, thank you very much. I'm looking forward to sharing this next hour with you. And so today we're going to be talking about microcystin and conventional treatment and using a multi-barrier approach. However, we will start with an introduction basically on microcystin itself before we get into it and a little bit of background about it. So down here in your lower right-hand corner, you will see a microcystin molecule, and that molecule, I'm trying to get my pointer. You'll see a microcystin molecule, and we have over what we call 160 variants of microcystin. So what we have here is what we call a cyclic hepatopeptide, and you'll see 1, 2, 3, 4, 5, 6, 7. So there's your 7 peptides, and in those we have variants, and those variants commonly we see different amino acids in the X position and the Y position. So common you will hear microcystin LR. So this would be a leucine arginine, or LA. So it'd be leucine alanine. Microcystin is a hepatotoxin, so its primary action is on the liver. It inhibits protein phosphatase, and protein phosphatase is actually an enzyme that that basically removes phosphorus off of the protein, which enable then the proteins to enact. So when you stop this process of removing the phosphates, it actually, the protein stops functioning. When the protein stops functioning, the cell dies. So that's the mechanism how it works. Later we'll talk a little bit more about protein phosphatase because we actually use it as an indicator. The toxin region of the molecule I just highlighted, so it's considered what we call the ADA group, and then the glutamate that's on the top there. So those two areas are considered the toxic region, and that's the area that binds in the pocket of the protein phosphatase. Over here we have the region in which it actually binds then to the chemically binds to the protein phosphatase. Unlike other things that inhibit enzymes, this one will chemically bind, which actually makes it fatal, which means there is no reverse action off of it. So what about cyanobacteria blooms in general? Well, we need to be able to identify them. If you look at the bottom, you'll see a Canon and Linka reference. This you can actually go to the website and pull it up, and it'll help you identify them. In this case, the microcystis bloom, oftentimes when people walk by it will think that green paint has been spilt over the area, and no, it's algae. This one commonly referred to as pea green or pea soup. Those two are microcystis. This one, you'll notice that they look like little horse tails or horse brushes, like tails going. This is a phanazominin, where the first two will produce microcystin. A phanazominin does not produce microcystin. Phanazominin, although it can potentially produce other toxins, it does not produce microcystin to date. We have not seen any. Dolichospermum is actually what we used to call anabina. And you can see it looks like a glob. And then finally, I kind of just give you an overview of Lake Erie from what we call the MODIS satellite. So if you want to look at blooms and you want to have an idea of what they look like, please look at this reference. It's a very good download for you. If we look here, now we're going to look at the types of microcystin producing cyanobacteria. We just talked about Microcystis aeruginosus. There's a few microcystis that don't produce it, but many of them, when you identify it as microcystis, do produce or can produce the toxins. Planktothrix is capable of producing the toxins. Anabenopsis is capable, and finally the Dolichospermum. Those are the most typical microcystin producers. And if you want to learn how to identify cyanobacteria and other algae, you'll notice down here we have a reference. And this is, again, is a download, and it's an algal key. It's a very good one with pictures. Actually, all these pictures came from that one. It's by Barry Rosen. So the next thing you might want to, that I'm going to talk to you about is, where is the microcystin? Well, in a healthy bloom, which is an intact bloom, we have 90% of the microcystin is going to be inside the cell. And so what we call intracellular, or some people call particulate, being held in the cell. Outside of the cell is what we typically call, which is going to be 10% of the microcystin, is extracellular or dissolved. Now this really matters from a treatment perspective, because if you precipitate it all out in your coagulation and you get a nice coagulant flock coagulation and it drops, you still have 10%. You need to know that. That's going to be dissolved. So we need to know how to treat the dissolved, and we need to know how to treat what's inside the cell. So it's extremely important, again, to know where the microcystin is so you can treat it properly. I call this on my way to regulation, or on the path to regulation. So here we have our highway. And yes, I was around a long time ago, and the CCL1. CCL1, from a historical perspective, was referred to cyanobacteria and their toxins. So they did two things. They basically included the cyanobacteria, and they included the toxins without listing them. By the time we got to CCL2, we had removed the cyanobacteria, and we had just started listing the toxins. So in 2 and 3 and 4, we see we don't see the word cyanobacteria. We don't see the word, I think you see the word cyanotoxins, but you see microcystin, you see anatoxin, and you see cylindrospermospsin. So those were the ones that were decided by an early on expert committee that would be of concern to the United States from a cyanotoxin perspective. Along the way, though, between 3 and 4, we had a health advisory pop up. We're going to talk about that, and we're also going to talk a little bit about UCMR, since I believe that is really important for you guys. So from a health advisory perspective, what is a health advisory? Well, here we're going to go with the technical. An informal, non-regulatory guidance for non-regulated drinking water contaminants to assist federal, state, and local officials and public water systems in protecting public health. So the two things to remember are these are kind of recommendations. They will give you a little bit of guidance, but they're not regulated. So every state's going to handle this type of announcement differently, and it's important for you to understand how your state handles it. You could go to one extreme, which is like Ohio, which they actually regulate it, and there are going to be still states that don't acknowledge it at all because it's an advisory. The other thing that came up new this year was down, you can see at the bottom, is I call it a two-step. We have a children's health advisory level, and we have an adult health advisory. So we're just going to look at cylindrospermospsin first. So if you look at the bottom here, you'll see ages. And if you look at the top, it's what we call the amount in the drinking water. So it would be microgram per liter. So one thing that we noticed over with children is we know that for body to mass, to drinking, how much they drink relative to their mass or their weight, they drink more in proportion. So in theory, they're going to take in more water per mass than adults do. So this is the reason that the EPA believes that you need 2 levels. So in cylindrospermospsin, like I said, occasionally we'll talk about the other ones. You see a 0.7, and then this is up to preschool age, and once you get to school age, you see it jump up to 3. For microcystin, you'll notice it's much lower. It's at 0.3 across, and then you jump up again to 1.6. Now remember, the WHO guideline is 1, so we're below the WHO guideline here at 0.3, but we're above the WHO guideline here, and similar to the Canadian guideline. So the next thing I said we'd talk about is UCMR. So yes, it is on list 1. So the contaminant cyanotoxin is on the upcoming UCR 2018. You see microcystin, anatoxin, and cylindro. We're going to actually talk a little bit about all of those and how we're going to do it. We're going to have to look at the source water and the groundwater under the influence. Our time frame is going to be between March and November. Our frequency is going to be twice a month and for 4 consecutive months in this time range. Sample events must occur at least 2 weeks apart, and you're going to have to do it over a 3-year period, so between 2018 and 2021. Anatoxin is pretty simple. This is going to be an entry point into the distribution, so it's going to be at the end of your clear well, typically. We're going to use EPA method 545, And it's what we call a shoot and go. So basically, when you send your sample in to be taken, they're just literally going to pull some up into their syringe and put it into, a calibrated LC, liquid chromatography, mass spec, and they're going to run the analysis, and they're going to get their analysis out. Microsystem, on the other hand, is a little more complicated when it comes to the upcoming UCMR. You're going to grab 3 samples, and so the 3 samples you're going to grab them, they say at the same time. You're going to grab an intake sample, and then you're going to grab 2 entry points into your distribution. So what are you going to do with those 3 samples? Well, you're going to take your intake water, and you're going to take your temperature of the intake, and then you're going to come down, and they're going to run ELISA on that. And you're going to take your other 2 samples that you took, and you're going to put them in the refrigerator. And then you're going to determine what your ELISA concentration was. If it's less than 0.3, then you're done, and that's the value you have to report to EPA. If it's more than that 0.3 level, then you got to step to the next step, which is you're going to have to do an ELISA on your finished water. When you do your ELISA on the finished water, if it's below the 0.3, you're done. You send the result in. If it's above the 0.3, you're going to basically send your next sample in, which is for your LCMS, and get analysis done by LCMS. So what are these 2 methods that they're talking about? Well, on your right-hand side, you'll see a box. That's an ELISA kit box. That is a Brexis ADA kit. And the method that people will be using is EPA method 546. And below you see an LCMS-MS, and you'll be using US EPA method 544 for Microsystem. It's important for you to understand the methods and the limitations on these, and they are actually very in the QA/QC on them. So we can discuss that later once we get through if anybody wants to talk about that. How does this ELISA work? Well, this is what we call an indirect competitive ELISA. The conjugate is independent of the ADA for detection. So what we have is the black dots are going to be your microsystems. The blues are going to be your antibody. And then down here we have what they call the coding antigen. So the antibody has a choice. It can bind to the microsystem or it can bind to the antigen. And basically you get what we call equilibria. So the more black you have, the more of the antibodies are going to bind to the black and the less that are going to bind to the antigen. And so at that stage, when we have everything bound, so the antibodies, the black is bound to the antibodies, we actually rinse the case. So you rinse it. So you actually rinse the microcystin away, leaving behind just the antibodies bound to the coating on the bottom of the plate. And that's what you see here. Then you add in another anti to the antibody, and it binds. And then that little thing up here that you see is what is going to be in what we call the enzyme that actually makes the color. So the thing to remember on this, if it's the first time you're using it, the dark color is low numbers and the light color is high numbers. So it's kind of the opposite of what you would think, but that's how the kit works. So kind of in a nutshell, what I would like to do is for a minute talk about one of the problems that we've seen. And one of the things that we've seen is the permanganate, and when we use the ADA ELISA kit to study the microsystems. So in this case, I'm going to step through this hopefully slowly to make sure I don't lose anybody. So we started with the 50 part per billion microcystin LA. And when we started with that concentration, we got these values at 0. So we have basically, here you can see what we did was this is a ratio to TOF, which means this is going to compare the ADA kit to the mass spec results. And then I'm going to compare what we call a PPIA kit, which is using that protein phosphatase inhibition assay. So we actually use the protein phosphatase to detect it. So these are 3 methods, again 3 methods: protein phosphatase method, an ELISA method, and a mass spec method. And we're going to compare both of them to the mass spec because mass spec has all the quality control that we need. So if we start at this level, and this is 50 part per billion, and we take the PPIA results, which we get as 50 part per billion, and divide it by the 50 part per billion by mass spec, we get a number close to 1. But if we take the results from the ADA kit that uses LR, we get actually a result closer to 2. So basically, when we have the 50 part per billion in the kit, if we do it relative to LR, we see twice the response. As we progress through time, we are reacting with permanganate, and you can see that the reaction is relatively slow. We don't see much of the reaction going on. However, up here, I shouldn't say going on, we see the mass spec responding the same way to the PPIA. Again, the mass spec responding the same way to the PPIA. And we actually see, as it goes on, we see the concentration go down. But what we noticed here is if we stayed at this ratio of 2, we would say it's responding the same way. There's not a problem. But at about 10 to 20 minutes, what we see then is that ELISA starts to respond differently. And we actually have a false positive going on where it's responding, the ELISA is responding to a byproduct of the permanganate. This is a problem because if you actually are using permanganate, ELISA is not the best way to determine what's happening to your microcystin. Either you're going to use a mass spec or you're going to use a PPIA kit, which you can purchase. Okay, so from a tools perspective, if you're going to use permanganate one more time, you're going to want to try to get mass spec results, or you're going to use a PPIA on site. Or you're going to be elevated and you're going to get some false positives. So optimization of, the next thing we're going to talk about is treatment. And we're going to talk about the optimization of conventional treatment processes to reduce, in this case specifically, microcystin. We always are going to use a multi-barrier approach. And each barrier, remember, is important and plays an important role. So even if you don't move 100%, the bottom line is at the end of the day, when we get to the end of the clear well, we want no microcystin going into the distribution. So even though some of the barriers may be less, remove less, they still can be important because when you're playing a part of that big picture, you are taking things, you're removing them out. So again, the combination of barriers is going to give us resilience, and we're going to look at those barriers now. Remember earlier we talked about extracellular and intracellular. Remember extracellular is outside the cell, intracellular. So if we look at some of the control methods, we're going to say yes, we can remove extracellular and intracellular up front. And those methods are going to vary. We're going to talk about them. Preoxidation, yes, and we try not to remove the intracellular. Physical absorption, we'll talk about methods there. Yes, for the extracellular, no for the intracellular. Conventional, no for the extracellular. In other words, coagulation sedimentation, your extracellular is going to go right through it. Yes, and we're going to talk about how much that varies. And disinfection, yes and no. So by the time we get to disinfection, one of the first things I'd like to spend a minute on is early warning. You know, what can help me determine the potential of what's coming ahead? So is there something that can give me a little bit of a warning? Well, one of the things that people talk about is the images from the satellite, and we're trying to work on them. But I'd like to remind people that people have cloudy days. So this is actually a week before we went into the Toledo crisis, where they had a huge bloom. And you can see that the cloud cover isn't showing you much of what's going on because you can't see and hear what's going on. And then the next one, again, cloud cover is distorting here by the intake. And cloud cover again. So meanwhile, while the operators are watching this to get an idea, now they're getting an idea that there could be a problem, because even though they've got cloud cover in here, you're starting to see quite a bit of green, and it's coming through. Next image they look at is here. Now they know they have a problem, and actually they know they have a problem because they can see it in the intake, because you see the green involved here. And then now they have a problem, they know they have a problem, and they check to see if the problem's getting any better, and they get this. So notice we need to be careful, because images that we're pulling down, if there's clouds, we're not going to see what's going on. And then you notice I have clouds cause a problem, and you can't read it because the clouds are there. Other early warning systems that we consider good practice would be a buoy system with a fluorescent probe. Shortly after the Toledo crisis, Toledo put in two buoys that they monitor, plus they can use the other buoys that are there by NOAA. So those buoys actually goes with this data down here, which you notice is they have both a chlorophyll probe and a cyanophycin, which is specifically for cyanobacteria. And you'll notice in 2014, even though we had a lot of algae, a lot of blue-green, we had a lot of toxin, you notice that it doesn't really flag. So this buoy went in about 2 weeks after their crisis started. Now, mind you, their problem went for clear into October. So they actually were using the buoy for the last 2 months of their problem. But what you'll notice is there's a lot more phycocyanin in 2015. So they had a lot of cyanobacteria in 2015. They had very little toxin in 2015. Another way to do this is to bring in cell counts. So for a long time on the Ohio River, we would count basically blue-green algae and all the other algae. We'd plot them according to what they were from a drinking water perspective. So we plot the ones in taste and odor and toxins, and then we'd give people weekly counts of what's going on. So if things started to climb up, they could see it, or if they were coming down slowly, they could see it, or if they're in the flat period, just to give people like a control chart on it. And then again, we talked a little bit about the Microsystem, but there's a beautiful picture that gives you a great idea of what's going on. Okay, so we're going to now step through the process. So a standard conventional treatment plan is, I usually break it up this way: source water, preoxidation or screens. So people use preoxidation for several reasons, and they can also hinder or help you. We're going to discuss that. We're going to talk a little bit about coagulation, flocculation, sedimentation, filtration, and then disinfection, and where it can help you there. And then once it's in the distribution system, we're not going to discuss that today. Source water. So how can we deal with source water? Mostly I think about it as an intracellular perspective. Depending on what you have for your source water, whether it's a river, a creek, or a lake, or a big lake, or a small lake, will depend on if you can use any of these things. But out there, we know people that flush, so that literally if you actually, like, nobody would do this on the Ohio River, just so you know, this is just kind of an example. But if you're on a river with multiple dams or small dams, potentially you could open a dam up and push it on down. We do know of people that have pushed it down to the next drinking water plant, which is kind of interesting, but you can open the dam and flush it. And then you can potentially harvest. There are harvesters out there. There are people that will come and harvest for you. You can divert. Maybe you have 2 intakes and you can take it from the second intake. Some people add flocculant up front and do coagulation. Some people aerate. Those are usually aeration, mixing these 2 things, and algicides. These are usually something that is more done, I call it, on recreational lakes, and done, not that they can't be done, but this is usually a group idea. So it'd be the drinking water plant, recreation, and other things, other people talking to get to that. Intake. Some people do have a little bit of control at their intake, but it depends on how easy it is. Some people have variable intakes, so they can actually adjust the height on which they're going to be at. Remember, many of these cyanobacteria will come up and down the water column, so sometimes you can move around them, and sometimes you can just get deep enough down that the algae never go down that low. Now, there are other issues of pulling in deep water, but it's all a balancing act. So you need to know your water supply. You need to know how easy it is potentially to take at different intakes, and potentially look at that as an option. As you adjust it, you're going to be adjusting basically intracellular. You also might determine when you take your water. So some people have storage reservoirs, so that if they pull the water in at night because the algae is not at the bottom at night, or they pull it in during the day, depending on the diurnal swing of the algae that's there. We're going to talk a little bit about extracellulars and 9-pack on the next slide. Conventional treatment. The general rule of thumb is you really want to keep those cells intact. Remember, 90% removal by good removal of the cells is really important. And here's to give you an idea, and this is actually, I like this slide because this was actually Toledo, just a week after their problem. One thing that you'll notice is that they're in a bigger lake, and in this case, in a deeper intake. So there's 2 things. You can see it. We have the dark one here is a total microcystin, and the little one down here is the dissolved. So you can see the dissolved here is about 10% of the total, and so that's good. That's their bloom must have been pretty healthy. But the interesting thing is that you'll notice that we have spikes decrease up and down, And that's very hard to treat. So shallow lakes, oftentimes rivers that are acting like ponds, you won't see this type, and you'll be like a constant intake. But it's important to know, because if you're pulsing different levels of cells, you have to be aware of that, because your treatment can't pulse this fast every 2 hours. So oftentimes you have to kind of adjust to allow for that pulsing. All right, so the next step is coming in is preoxidants. Why do people use preoxidants coming in? Well, people, first of all, why do they use them? Well, they use them for zebra mussels. They use them to remove the manganese. They use them to keep their inside of their intake away from other things, maybe besides zebra mussels. But zebra mussels and manganese seem to be the two major ones. The preoxidants can vary: ozone, permanganate, chlorine, chlorine dioxide. I'll have a summary slide in this in a minute. Ozone will lyse the cells, even at low levels. Chlorine will lyse the cells. Chlorine dioxide will not lyse the cells. Permanganate, there's been a lot of studies showing that low doses of permanganate does not lyse the cells. All of these, except the chlorine chlorine dioxide, will react with the extracellular or the dissolved microcystin. The problem is again, you want to keep it intact. The other way you can remove dissolved microcystin is by adding PAC, powder activated carbon. So oftentimes what we see is the preoxidant followed by PAC. Again, you want to control your cell lysing. You don't want a lot of cell lysing. So here's a summary slide that's pretty important to think about. So the extracellular toxins, so we have the extracellular toxins. So chlorine for microcystin, we're not going to talk about these guys, we're just talking about microcystin. Yes. Ozone, yes. Chlorine dioxide, no. Permanganate, yes. So these three will react with the microcystin, removing the toxicity. Chlorine dioxide, no. On the other hand, adding chlorine up front and ozone up front, you're more apt to lyse the cells, and you're going to release that 90% inside of it, and then your demand will decrease, and you'll probably still have leftover toxin. Chlorine dioxide doesn't lyse the cells, but it doesn't chemically react either. It doesn't inactivate. Like I said, there are 3 studies that show that the permanganate can be added at less than 2 milligrams per liter, or 2 ppm, and it's not likely to lyse the cells. There are 3 publications that suggest this. There is some more recent research done by EPA saying it's not. I'm going to say, though, the 3 publications that say it's possible use LC-MS to determine microcystin removal, whereas the other one uses the ELISA kit from EPA. The powder activated carbon that we use typically will be wood-based. And the fact that it's wood-based is because we'll talk about pores in a minute. The pores are larger. Microcystin is a 1,000 molecular weight. It's a lot larger molecule than your taste and odor compounds. If you want to know how well your PAC works, you can do it via jar test. And then you can also do other things as you can use a jar test, which means you add microcystin, you add your PAC, and then you pull it out and see how much microcystin is left. We do not recommend pre-chlorination before PAC, because also pre-chlorination before PAC destroys the PAC. This is a busy slide, so we're just going to kind of step through it, and it does a little bit before and a little bit after. But coagulation sedimentation, I'm going to start on the right-hand side. Some of the monitoring tools that you might want to look at. First of all, turbidimeter is usually not a good sign for algae, because turbidimeter works on scattering light, and algae absorbs light, sort of like carbon does. So turbidimeters don't work well on carbon, and they don't work well on algae. Current stream detectors, you will see a negative. Algae has a negative charge on it, so you can actually see that change as you pull in an algal bloom. Particle counts you can use, obviously chlorophyll or phycocyanin. You can use counts, and you can use the ELISA for pre-screening to give you an idea of what's going on. So over here, when we talk about coagulation sedimentation, basically you've already done potentially some pretreatment or not, and you're going to add coagulation, sedimentation, and you're going to just basically try to remove the cells. Your goal to remove the cells, in general, the rule of thumb is try to get less than 100 units of algae per mil to go on top of your filter. This will guarantee you longer filter runs. If you don't do a better job at coagulating, settling in the settling basin, then potentially what you're going to have is your filters are going to start to clog earlier, and you're going to have more backwashing going on. From an extracellular perspective, we talked about PAC, but remember granule activated carbon can either be in your filters or can be put to the top of your filters to help remove the microcystin that's not within the cell. So how well can we do? How well can we remove cyanobacteria? Well, we can remove cyanobacteria really well. This is what's out in the literature right now. So typically we can remove about 99.5 if we're just under regular coagulation sedimentation. If you're looking at line precipitation and you optimize your system, we're talking 99.5. Ballast flocculation, which is a much faster process, we're looking at about 95% removal. And coagulation sedimentation on microfiltration or ultra, we're talking typically 90% removal. One of the studies that was reported by Schlag, and it was done, you can find it in Toxins, is basically these are conventional treatment plants. You can look at what they're adding. And we wanted to know how many cells were removed percentage-wise. So we have log removal of both cyanobacteria, and then we call them potential toxin producers, cyanobacteria. And you'll notice that the numbers are very similar from side to side. One thing that you'll notice, you said we only got 0.2 removal from Oklahoma at this stage, but that was because there wasn't many cells to begin with. So this is not only during high cell time, this would also be, or bloom time, this would be in general throughout a summer. So the 0.2 levels meant that there was not a lot of cells coming in. In general, this study showed that most drinking water plants removed cells to under 1 cell per mil coming out of the bottom of their filter. This is what you don't want to see. So this is what we call a very bad day. So we talked about algae moving up and down the column, and we talked about coagulating it. If we coagulate it well, it grabs and it sinks, and it doesn't resurface. If you don't do a good job coagulating it, you're going to have it floating in the top as you see here. So we would call this cyanobacteria breakthrough of the clarifier. Zamati has a lot, 2 publications on this, and they're listed down here. And they did have high levels of microcystin at the top of that. So filtration, conventional filtration, we're going to talk a little bit about conventional filtration, biological activity, GAC, and low pressure membrane. Actually, I don't think I talked about low pressure membrane because that's a conventional plant. Biological filters, what do we expect to see from them? We expect to see the intracellular toxins stuck to the top, but we also, anything that was extracellular technically can be removed by biological activity, and we do actually see filters that are biologically active remove LA, LR, cylindrospermospsin, and anatoxin A. Oftentimes we do not see it removing saxitoxin. We see the removal of it in everything from slow and fast and rapid, I should say slow and rapid filtration. And typically the removal can be as much as 15 or 20%. GAC is going to be absorbed onto it. And one thing to think about is it's a very effective as long as your filter bed, if you're using absorption and not biological end of a GAC filter, it's very effective unless your pores are all filled. So you have a high TOC count, and then you have a let's say your bloom. If your TOC is filled into your GAC, then the LA or LR can go through. Another thing to think about is that we do in the literature see a difference between the absorption of like LA and LR. And typically in our GAC that we see, because we tend to lean toward more of smaller molecules, the saxitoxin, the anatoxins are more readily absorbed because they look more like, or more of the size of what we would call taste and odor compounds. So what am I talking about in pores? So the micropores up here, we tend to think about those for the taste and odors, the industry, solvent spills, and anatoxin. So the micropores are the small pores down in here. And if you have mostly micropores in your carbon, you're not going to do a good job removing microcystins out the microcystins. So the mesopores, the mid-sized pores, you're going to do better at removing the microcystins, the cylindrospermospsins, and the anatoxins, which again are a little bit larger. The other thing that you'll notice is, in general, people think of the RR absorbing better than the LR and the YR, LR, LA. The larger pore, again, seems to be more effective for the larger amount of microcystins. So what we're looking here is, so let's go back for a minute and start at the beginning. So what if we started at the beginning here? We have phantoxaminin, we have anabina, we have microcystis, and we have pseudoanabina. So this is where we're starting at the beginning. So we bring it in, and we maybe add permanganate. The cells don't lyse. We remove some of the toxins, the extracellular toxin. We add PAC, and we finish up what we didn't react. We remove from the extracellular toxin, we remove with PAC, and then we go into coagulation or flocculation, coagulation, and settle it out. And what we notice is the microcystis, the pseudoanabina, planktothrix, anabina down here, which is the dolichospormin, these tend to precipitate out better than the phantoxaminin. So the phantoxaminin can come on top of your filters, and then what we noticed is on our study and somebody's study, we saw if phantoxaminin if we saw anything break through, it tended to be a phantoxaminin. This is kind of unusual because they're long strings, and all we can think of is that you get individual strings and they can just wiggle through the filters. But again, none of us in this process, none of us saw high levels of phantoxaminin, but we did see a phantoxaminin coming through the filters at very low levels. So let's talk a little bit about disinfection. So let's say we have some residual toxin at the end. What can we expect? Well, we can chlorine. Chlorine is going to be most effective. The closer you get to pH 7, the more effective it's going to be. The further you get away, for example, pH 9, the less effective it's going to be. That's just working on the basis of how chlorine disinfects. The closer you're to 7, the more oxidation potential power it has. The farther away, the closer you are to 9, the less at least it has. It's the same with the microcystin molecule. So we're pH dependent. We're going to be temperature dependent, and we need to think about that as we're going into our clear well. A lot of people have adjusted their clear wells to be between 8.5 and, I hate to say 9.5, but I have seen them up that high. And for maybe something like lead and copper to actually help maintain the lead from coming off your pipes. When it comes to oxidation power, you don't have it necessarily to now work on the microcystin molecule. There is what we call Cyanotoxins. AWWA has a online program that can help you predict how well your clear well will work. Then we have ozone. Ozone is wonderful. It reacts very actively with a set of double bonds here. It's extremely reactive. We have chlorine. No, not in the clear well. It's not going to help you. Chlorine dioxide, it's not going to help you. The hydroxyl radical, yes. Permanganate will help you, but you usually don't add permanganate to your clear well because you will end up with pink at the end. Usually permanganate's used at the front of the plant. So in summary, we can talk what I would like you to think about and maybe ask some questions if it wasn't clear. I would use the ADA ELISA as a screening method. If I'm looking at how well my permanganate's working, I would not use the ADA ELISA. I would use either PPI-8, or I would set up with a lab that they knew my samples were coming in that day, so they can run them overnight and you can get the results the next day. So I would use LC-MS-MS. Again, you could use ADA for screening, but I would only, as an analytical method, I would use LC-MS-MS. US EPA has posted health advisories, and it's a 2-level health advisory, which is new to us. It's preschool versus adults and school-age children. you might want to think about how you're going to do that. In the past, we usually just get one level, and it's that. There are people talking about using it as a 2-level. There are people talking about using it as a 1-level and just sticking with the preschool level. And just basically that would be their warning or their go-out to. Remember, with those, you do have a 10-day time period where you can work on your treatment and get it down. So you have some time to it for treatment adjustment. The unregulated contaminant rule is going to come in 2018 through 2021. And remember, the microsystem is going to be a little more complex when it comes to grabbing samples, and you have to make sure that you know the order in that's going on. Remember that a lot of us have been treating algae for a long time. We actually do a really good job in conventional treatment, removing it. We don't want it on the top of our filters. So if we keep those practices in line, coagulation filtration works great at removing intracellular. Extracellular, you're going to depend on absorption and oxidation. And on that, we can open it up for questions.
All right, Judy, thank you very much. We're going to be checking for questions right now. I know we do have one comment that came in. In the meantime, I just want to say that there are a number of ways, if you have questions later on, there are a number of ways for you to contact us. Here's a diagram that shows our field salespeople and their telephone numbers. So if you want to contact somebody in your region, feel free to use these numbers as well. If we have anyone who is online from Europe, or if you have any contacts in Europe that might like information, we also have our in contact list shown here as well. And then finally, here in Illinois, we also have contact information that we can provide. My name is here, as well as Sean Davenport's, if you have any further questions. So Judy, we have a great comment here, first of all, from very early on regarding the UCMR requirements. And the comment is that the final proposed, or the final rule, will include probably finished water monitoring only and not source water. So are you familiar with that at all?
I haven't checked recently, so that just might be that I haven't updated. I should probably go look, but the last time...
So everybody listening can check somehow to see what the current...
Right. Okay. Yes, and so I guess if that... Yes, I would argue you should check for the final. I did not know they had a final out yet, but I will check and update. Sorry about that, anybody?
All right, and just as one quick reminder to everyone attending, we will have a recording of this available. We will email all attendees and registrants the link of the recording, either today or tomorrow. Please feel free to share that. Also, if you would like a copy of the PDF or be added to our email blast, please let us know, and we will be happy to take care of that for you. And if you do have any additional questions, please feel free to type those in. We will wait a few more seconds to see if any more questions come in. If you have any questions after the webinar, please go ahead and contact us, and we can get in touch with Judy as well. All right, great. We thank you again, Dr. Judy, for presenting on our behalf, and thank you everyone for attending.
Okay, thank you. Bye-bye.